Hello and welcome to this video about DNA fingerprinting. This is a process that is used in a couple of areas, for example forensics. So if you wanted to find out whose DNA matched a crime scene, you can use DNA fingerprinting to help you find out who committed the crime. Also in things like paternity tests, when you are trying to match the DNA from a child, the DNA of the parent, you can use a DNA fingerprinting to find out who the father of the child is, for example, just like you'll see on Jeremy Kyle or something like that. So what happens is they take a small section of DNA, for example, it might be something that they've collected at a crime scene, or it could be a swab of the mouth or something like that, and they'll have that DNA, and into that they'll just add some enzymes, which will chop up the DNA into little fragments. They'll then put those fragments of DNA onto some gel. So it'll look similar to things that you've seen in class before, like agar jelly or something like that. Just a gel consistency, and they'll put the DNA fragments into here. So it could be, for example, that in here they would put the DNA at the crime scene, perhaps in this one, and in here they would put the suspect's DNA. And what they're looking to do then is match the DNA that they found at the crime scene to the suspect's DNA. What happens when they put it on this gel is they run an electric current through the jelly, and that helps the fragments of DNA move and separate. So they use an electric current through the jelly, and you don't need to know these words, but you may see it if you go on to A level. This process is called gel electrophoresis, and it's a really common technique when you go into A level, and if you go on to study biology at university, you'll use it all the time to separate DNA. So here we've got the fragments of DNA, an electric current is passed through the jelly, and the fragments move across the jelly and give a certain pattern where you're going to get bits of DNA left. Now if we compare that, then it's a little bit like chromatography. When you're comparing the pattern with other samples, all you need to look for is where the same pattern is expressed. So if we look across the rows, you'll notice that suspect 2 here has the same pattern as the DNA found at the crime scene, so that is a match. Okay. Just one extra thing then, if you're looking at this in the context of a paternity test, obviously you're not going to get two completely matching sets of DNA because your DNA is not identical to your parents. It's a mixture of your mother's and father's DNA. So in a paternity test, what you would be looking for is a DNA strip which had similar patterns to your own. So obviously yours would be a mixture of your mother's and father's patterns, and if you're looking for your father's DNA, his pattern of DNA fragments will not be identical to your own, but there will be several parts which are identical to yours. So it's called fingerprinting then, because just like your fingerprint, your DNA is unique to you. Not unless you're identical twins, you will not have the same DNA exactly as another person. If you found this video useful, then please press the like button and feel free to subscribe. Thanks for watching.